UCLA, giving us a background on the viral and eukaryotic communities of urban ecosystems across U.S. metropolitan areas. All kingdoms of life deserve some love, or at least scrutiny, so we'll hear a little bit more about the euks as well.
Great, thanks. Thanks for organizing. So it's a great opportunity to share some new results we have got, and then I guess like we took. So I guess like we took the message from David. So where and Pavel, where the motto of CAMDA is like to explore the data in the way you want. I think it's like very unique opportunity, which you don't have usually. You have strict plans and stuff like that. So we decided to do something which is usually ignored. So if you think about microbiome, so that's not really the bacteria only. And then every time I see a paper claiming to study the microbiome, and then if you look inside, there is only bacteria results, make me upset in a way, but also it's a nice opportunity to say, hey, that's great, so we can explore fungi, eukaryotes, and the viruses. And so if you think about the microbiome overall, there is other kingdoms of life which are not only bacteria. And I think I should put the quote of Chris here, just good one. Okay, so if you think about the metagenomics, so that's not 16S, and then again, there is a big confusion in the field. So many studies claim to do metagenomics, even they do only 16S. So this is not that. So really, like metagenomics is really about just whole genome sequencing on the sample of the environmental or urban sample. And if you compare 16S with the metagenomics, which is really whole genome sequencing, then you will see the certain advantages of metagenomic. Namely, you can go beyond the bacteria and profile other kingdoms of life. And that has a little bit to do with the methods which are the marker-based methods, which even giving metagenomics, they are limited to the bacteria, or they have very limited reference from the other eukaryotes. So our idea was that, so first of all, the metagenomics, so just like giving the background, so you got your sample from the urban microbiome, from the urban environment, or like just the normal environment, and then you will get many reads, and then they are coming across all the kingdoms of life. So the data we have got was the CAMDA challenge, where we got the New York, Boston, the Sacramento, and then we managed to run the whole Boston data set, the whole Sacramento. We managed just run very few New York stations, and then we are scaling up, so it's running, so the results will be soon. And then I guess as we look to the data, I think one challenge was that all the data sets except Boston were pretty much the same depth of coverage, except, so for the Boston it was quite challenging. So some sample were really few reads available, order of less than 1 million, and then other were really deeply sequenced. And then I will talk about the effect of that. If you think about the metagenomics overall, then one challenge is that there is so much unknown. So if you compare human studies versus metagenomics, so usually for human, you will manage to map the majority of your reads on the human reference, so usually more than 80 or 90%. So if you think about metagenomics, that the numbers are much less, and then where there is around 50% of the organisms which are known. So one of our goal was that if we use the most comprehensive reference we can ever got, so can we go more? Can we explain more reads? And then I don't have a slide for that. It's still, we're dealing with many caveats there, but then we were able to increase the fungi, the eukaryotes, and the viruses. I think we didn't increase the viruses, but we really increased the eukaryotes and the fungi by order of like 5% or so. And then before we'll talk about our method we developed, so I think if you think about the existing methods, they primarily focus on the bacteria and archaea, and then they are either marker-based, so you basically have the marker genes which are considered universal, present in a single copy, or those are the k-mer base. So you generate the state of k-mers, and then if the k-mer is present, if the k-mer from your read is shared with the k-mer generated from the certain genome, you will say, hey, that's a match, and then you will report that species as a present. So there is certain caveats of using those methods. So first of all, if you think about the k-mers, so if the genome you're observing in an environment is divergent from the reference, so the k-mers might fail. And another thing that I guess that there is nothing in between your sample and the relative taxonomic abundances you have got. So where we think that should be something in between, where maybe the user can make a decision if he thinks there is, or she thinks there is enough evidence to conclude that a particular bacteria strain or the fungi is there. And then I guess like I don't want to get too much controversial, but then for the what is suggested to be failure of bioinformatics, if you use only the marker genes, you should really think about the homology of those marker genes to other pathogens. So I guess for the New York, it was a big booth where they detect the dangerous bubonic plague, the bacteria causing the bubonic plague. And then the reason for that was that the marker genes, which was considered to be present uniquely in that bacteria, was actually shared with some non-pathogen bacteria. So I guess our idea is to utilize the coverages across the whole genome, and then actually we're trying that right now. I don't have results yet, but then our idea would be to take that genome and try to map the reads across the entire genome. And of course, you will have uniquely mapped reads being mapped to these marker genes, but we also expect the reads to be kind of nicely mapped across the entire genome, even being multi-mapped reads. And then we want to use the most comprehensive databases possible, and then we don't see the reason not to do so. So we acquired like the most comprehensive bacteria, viral, and the fungi and the eukaryote database with the many strains and the many genomes being available. So if you look at the EUPUB database, which is composed of the fungi and the eukaryote genomes, so we got under 100 different genomes per different phylum or like the kingdom as they're here, and then they are actually composed of the very many contigs, which suggests they are not so which we just merge into what we call the species. And then we developed the tool which we call NYCOB, which stands for Microbial Communities Profiling, where the tool is mapping based. So basically, you got your reads, you got your the reference, the most comprehensive you can get, and what you do, try to map. I think it's like, as if you look in perspective, so how the microbiome studies have started. So those people did map the reads on the genomes, but that type of the analysis was abundant for the reason just because the reference database were becoming larger and larger, so it was not feasible to map the reads on the genome, and then it was taking a lot of time, a lot of computational memory. So what people start doing that, they start doing k-mer-based approaches or marker-based approaches, which is suggested to be much faster. So our idea is, instead of mapping to the entire set of the reference genomes, we would rather select the genomes which are there based on the k-mer-based, any k-mer-based analysis. So the idea would be to generate the state of the k-mers from every genome, and to generate the k-mers from the reads and try to find the match, which will be really fast. So in this case, we don't care about the false positive. We are fine to include them. We're just really concerned about false negative, not to miss that. So our experiment suggests that, for example, given we're trying for the bacteria, and the viruses and the eukaryotes, so we see significant reduction in the number of genomes which we can pre-select. So, for example, for bacteria, you're having 1,000 or order of 1,000 of 10 of 1,000 of different bacterial genomes. So, doing that approach, you can narrow down to several thousand, which is really helpful, and then really speed up your mapping-based thing. And then after we pre-select the genomes, we do the mapping using any tool. So we find out that just running BWA actually is quite fast and also quite accurate. And after that, we try to utilize the multi-map trees, which are usually ignored. So if you have a read mapped to several different genomes, you will usually ignore it just because due to the structure, you have some homology between the genomes, and then the treat will be identically mapped. So what we suggest to use the so-called unique reads, with the reads which are uniquely mapped, which are only mapped to the particular genome, estimate the relative abundance of every genome based on the uniquely mapped reads, and then do kind of probabilistic assignment, where if you have a genome 1 with the frequency 90% and genome 2 with the frequency 10%, so you will say, hey, it's more probable I will assign this multi-map reads to the genome 1 just due to its higher relative abundance. So this way, this will allow us to kind of rescue the multi-map reads, which are currently ignored. So after we got the mapping, we trying different filters, basically just map based on the mapping profile. Can we confidently say that the particular genome is there? And then we use several criteria for that, and then I will discuss them in details in a second. But like overall, the idea is like how many mismatches do you have with your read, and the reference. Is the read uniquely mapped? Is it multi-map within the genome, or like it's multi-mapped across the genomes, and uniformity of coverage? So quite many metrics. And then also we, as the part of the tool, we want to generate a report for every species, where we will kind of say those things, as well as like providing the coverage plots, so that the final user can change the stringency, and then to adjust the stringency and use only the species which he's trusting based on the reports we generate. So I suggest you to look at the data. So this is what we are having fun with, just like opening the raw data and having a look. So this is the particular read which was mapped to the Malassezia, which is like the human skin fungi, and as well as it was mapped to some other fungi. And then this is what we call multi-map read. So in this case, I think it's possible to distinguish those two reads, just because I think the portion of the read being mapped to the first fungi was larger than this one. But often that's happening that they will be absolutely identical. So we will consider now only the reads which are distinguishable, meaning that there is a better mapping to a particular genome. And then, so we will consider only what we call the uniquely mapped reads. So if we look further for the BAM file, inside the BAM file, so another important parameter is like what portion of the read is being mapped, and then the soft clipping, meaning that you cannot map the beginning or the end of the read, is due to some adapters, so which you can potentially filter beforehand, but even not filtering those, or just like running BWA, is also, I guess, fine, where you will just observe some soft clipping. So why it's important, just because if the portion of your read is being mapped is short, then it's not that trustful, just because it could be some homology between the species or between the kingdoms. So as longer the stretch of the read is mapped, as more confidence you have. Another important thing is like number of the edit distance, right? The number of mismatches and insertions and deletion between your read and the reference. And then here you can see that it's quite a lot, even though the stretch of read being mapped is 100, but really there is 11 mismatches between the read and the reference. So we try to present the data in a way where I'm sorry, I think it's not the best quality, but then so this is the portion of the read being mapped, and then this is the number of mismatches, or like in other words, like edit distance. So what you can see here is, I think this is just default of BWA, where, for example, if the reads is quite short, then it will not allow many mismatches. So what we are most interested in is like what we call high fidelity reads, which the reads which are long with the few mismatches. So those reads we kind of sure that they're coming from that particular genome, as those ones might contain some false positive. And then we also look at the coverage across the entire genome, so we want the genome to be kind of uniformly as possible covered. So we don't want only one portion of the genome covered, but others are not. And then I want to show the example where what is interesting, so you have two different fungi, and in one you have kind of similar profile if you look at the reads, edit distance, and the portion of the read being mapped. But what is different, interesting, that here is lacking any reads, kind of high fidelity reads which are long, as well as like with the few mismatches. So our idea, the intuition behind will be to kind of trust this profile, but and not trust this one, just because we will probably, even though that could be true, but then lacking the high fidelity reads, we cannot really kind of reliably report this particular fungi. And this is one example where we try to put the thresholds, how many high fidelity reads are enough. So the orange line is the number of high fidelity reads, and this is the log scale. And then the blue line, the total number of reads, meaning from 30, the portion of the read being mapped is from 30 all the way to the read length, and the number of mismatches up to 15. So what you can see that there are certain fungi genomes which are probably false positive, where you're completely lacking the high fidelity reads. So we will definitely not report that, but as well we need to come up with the threshold, so we try to be conservative, and then we require to have at least 50 high fidelity reads, which we define the reads where the read length being mapped is more than 70, and then will be up to 3 mismatches with the reference. So using this approach, we look at across all the U.S. metropolitan areas, Boston, Sacramento, New York, and then what I report here. So this is the number of whole genome reads, I mean the metagenomic reads being available, and that is the number of fungi reads reported. So what you can see, I guess first of all you see the difference in number of reads, which are available for every city, where Sacramento is really deeply sequenced. New York is really uniform coverage, around 1 or 2 million reads, and then Boston is all the way from very low coverage data to quite nicely covered. So I guess as we will doing the analysis, that should be adjusted. The best way we see to deal with that is to subsample, and I guess you want to subsample to the certain level, resulting to ignore certain low coverage samples. So if we look for, so this was fungi. So if we look for amoebas, which is another eukaryotic pathogen, so we see quite similar picture, where the high covered samples of Boston and Sacramento have quite many reads coming from those eukaryotic pathogen. The similar was for the viruses, where the numbers were much lower, meaning that you have not that many reads coming from the viruses, but the pattern were quite the same. So we looked, so I have just preliminary results to share about the urban virome. And then what we observe is that the virome, the viral species, are really train station specific, meaning that they are not quite shared across the station. So, and here I showed just one top virus reported for every city. So in New York, we didn't find much coverage, I guess, as you don't have enough kind of deep coverage or deep depth of the sample, you will the number of reads kind of which will go to viruses are quite low. So we just got 200. That was the best sample with 200 reads aligned across the viral genome. The good news is that it was more or less uniformly covered, and then with the certain number of high fidelity reads, so which kind of makes us confident to report this one. And then for the both Boston and Sacramento, you're seeing much higher number of reads being mapped. But what we observed that we see rarely the viral species to be shared across the station, suggesting that probably they are not resident of the train station. In contrast to the fungi, which are quite resident. And then what I report here, so if we take the fungi and then kind of doing the same plot where this is the total number of reads and then the number of species, fungal species being reported, so we can see that even though the Sacramento was much higher covered, then it didn't result in more fungal species being reported, as well as was interesting for New York, we still find several samples. Actually, we find for New York 37 samples where was at least one fungal species being reported. And then if we took a kind of the heat map where we take the New York, Boston, and Sacramento, and we report the percentage of the sample that particular fungi was found. So what we can see really the widespread of the fungi, where I also colored the fungi by the class, where the first is human, then there is mold, and there is gray mold, and then there is all plant-associated fungi, soil fungi, and some others. So what you can see that, I guess, like especially for the Boston and Sacramento, you can see the widespread of the fungi, and then for the New York, it was mainly missing, but I guess like due to the coverage. And what is interestingly, we didn't find in New York the one which was really expecting to find, the fungi associated with the human skin. So it was none. It was not present in any single sample, probably again due to the limited depth of the coverage, because they are both were quite widespread across both Sacramento and then the Boston train stations. Okay, so I'm ready to wrap it up. So I think it's like, so if you remember one message from the talk, so just like remember the bacteria is not the microbiome. So this is what I'm trying to argue. So there is really a need to use the most comprehensive reference, not to miss other kingdoms of life. And then you can view it as a way to squeeze the maximum out of your metagenomics data. So we have a tool which will be available soon to profile the non-bacteria species. And then we observe the virome to be really station specific, and then the fungi will be to be spread across the continent, and then we can even claim the typical fungi of the urban North American eukaryote. The tool will be available soon, so you can check our GitHub page, and then we hope that will be really fast by doing the tricks we are planning. And then the future work, we want to explore the homology between the kingdoms, and then this is what makes us worry, and then maybe we'll try to redefine certain results, because if you think about the short read length, so certain, like the read of 50 will definitely might be mapped both to the bacteria and the fungi and so on, so we'll take care of that, and then we'll look for the other technical parameters, and then actually we're planning to extend the analysis if we have the data from your city. So it will be great to make a global map of the fungi and the viruses across the globe. And then acknowledging that the project was led by David Kalitsky, and there is people from UCLA and Georgia State University being involved. Thank you.
Any questions from the audience?
Hi, very nice talk, thanks. With regard to the virome, and I suppose it's more of a general meta-sub question, would it only have detected double-stranded DNA viruses, or like how were the samples prepared?
Yeah, I guess Yeah, I can answer that. I suppose that. Yeah, it would likely just be double-stranded DNA
Okay.
viruses. There's a protocol from Swift that does single-stranded. It's their 1S prep, but everything there is pretty standard, either MoBio or Maxwell extraction, and then a TrueSeq or QIAGEN library prep. So it's very likely it's just what it is. Yeah.
Yeah. And just another one about the urban virome. Did you detect a crass phage, which is found to be very highly abundant in fecal viromes, in like any of the urban ones?
I think so. I mean, we didn't. Yeah, I mean, it's kind of in progress. We didn't look so carefully for the viral. We really see it's very sparse as you look across the stations.
Okay.
But
thank
also
you.
it's very hard because like you see those viruses which are covered like with 200 reads, so like something like very low number of reads, and then we're still working on like deciding how confident we can be in that cases.
Thanks.
And I can say for the upcoming, we'll talk about this afternoon, but for future, the Metasub Challenge, it will go across all the cities of the world, and also some will be deep sequence and other more standard, and then some long and short reads, so that hopefully address some of the technical questions. I had one quick question as well. It looked like in New York, though, since I'm from New York, there was
Sure.
a lot of plant fungi,
Yes,
would seem
which
more
was
in
surprising because
the-
for me it's like because Sacramento is the light
yeah,
rail.
yeah, that's what I would have thought.
Yeah, so I would.
Do you know if those came from outside stations, or that's across the whole data set?
Yeah,
I
that's
think?
across, and then also we just use 65. So we just
65,
yeah,
yeah,
so we
so-
just really need to scale across everything.
Some of the initial data we didn't put in the first paper, but it's seen as that samples closer to Central Park seem to show more plant DNA.
Yeah. So I
with
think it's like
blasts,
yeah,
and so it might be
that's on the list to explore kind of the geography.
where it is.
Yeah,
Very
to see
interesting.
if it correlates.
But there's also rumors that New Yorkers are all robots, which maybe is indicated by the lack of human skin. So it comes up. So it came
Maybe
up
the
in
surface. We also
yeah.
want to explore the surface,
Good,
I
good.
guess.
What
But also,
differences?
if like certain fungi holds on certain surfaces, like we can check for the Bostons.
Very good, very good. Good talk. Okay, thank you.